All right, hello everyone. I want to thank the organizers for inviting us from the Office of Research Quality at NINDS to come talk to you today about experimental rigor and transparency. I'm Devin Crawford, but I also developed these slides with Shai Silverberg in the office. So it seems like experimental reproducibility has become the topic du jour recently. However, we can't even hope to have reproducibility until we have experimental rigor as well as transparent reporting so we know what was actually done. But unfortunately, many details are missing from published reports. And so the scientific community doesn't actually know whether or not research was done rigorously or whether unconscious biases may have slipped in. And that's what I want to talk to you today, is a little bit about how unconscious biases can affect research. So I am an NIH employee, but I need to tell you that any opinions I express are my own and not those of the US government. And other than that, I don't have any conflicts. So as Francis Bacon recognized back in 1620, we all have unconscious biases that can affect how we view the world around us. As he put it, the human understanding, when it has once adopted an opinion, draws all things else to support and agree with it. And though there be a greater number and weight of instances to be found on the other side, yet these it either neglects and despises, or else by some distinction sets aside and rejects, in order that by this great and pernicious predetermination, the authority of its former conclusions may remain inviolate. So in other words, we tend to confirm what we already believe to be true. But how does this affect our research? Well, unconscious biases can lead to experimental biases that skew results. David Ransohoff wrote in this opinion piece that bias is unintentional and unconscious. So we're not talking about intentional biases. We're not talking about misconduct here. It's defined broadly as the systematic erroneous association of some characteristic with a group in a way that distorts a comparison with another group. Went on to say that it can't be addressed by a simple process. Instead, it's complicated and involves making everything equal during the design, conduct, and interpretation of a study and reporting those steps in an explicit and transparent way. And so the first part of that phrase probably seems self-evident, but as far as reporting these steps in an explicit and transparent way, we need to recognize that there will always be unknown and known confounding factors in real-life experiments. So therefore, reporting the details is especially key. Now, to help with inadequate reporting in human clinical trials, the CONSORT, or Consolidated Standards of Reporting Trials statement, was first released in 1996, but has been updated a few times since then. And it contains 25 items that should be reported for all randomized controlled trials. And many of these pertain to experimental design and rigor. And it says right there in the CONSORT guidelines that randomized trials can yield biased results if they lack methodological rigor. And to assess a trial accurately, readers of a published report need complete, clear, and transparent information on its methodologies and findings. And so I need to make the point here that the CONSORT statement is not telling researchers how to do their studies, but rather is trying to help them transparently report what they did or did not do. And here I'll be listing some of the items in the CONSORT statement that can help determine whether or not there might be a potential for bias. First of those is how sample size was determined, not just what the sample size is. The type of randomization to put patients into different intervention groups and the details of any restrictions, like was it a block randomization? Who was blinded to the treatment groups after assignment to these interventions and how, as well as any losses and exclusions after randomization together with reasons. So why might data have been taken out of the study? And so all of these things we should be considering anyway to improve the rigor of our studies, but at the very least, we need to be reporting whether or not we did them for full transparency. But how do these affect results? Well, let's first look at sample size. This is a study of 660 meta-analyses from mechanistic human studies in 3 different disease areas: psychiatric, neurological, and somatic. And what they did, they took the overall effect size found in each meta-analysis and then looked at the underlying power of the studies that made up each meta-analysis. And usually studies try to aim for at least 80% power to detect the difference between groups. However, as you can see from this histogram, about half of the studies didn't even reach 20% statistical power. And why are underpowered studies an issue? Well, in the same study, they found a negative correlation between the size and the effect size, meaning that underpowered studies seem to have inflated effect sizes. And we can also find inflated effect sizes in other contexts. For example, if assessors within a study are not blinded. This here is a systematic review where they had blinded and unblinded assessors within the same study. And in these clinical trials, you can see on the right the meta-analysis. They found that 63% had a more optimistic, i.e. a larger effect size, as determined by the non-blinded assessor. That altogether, all of these studies created a total exaggeration of a pooled standard deviation of 0.23. So if we haven't taken efforts to blind ourselves or otherwise reduce bias, we could end up looking for what we want to find. And in preparation for this workshop, we wanted to look at studies more in this field. And so here is a Cochrane review, a recent one, on ketogenic diets and drug-resistant epilepsy. And what they did as part of this systematic review is they did a risk of bias assessment. And what they found for the 17 studies from 13 randomized controlled trials, in green is a low risk of bias for that particular item. Yellow is an unclear risk of bias, and red is a high risk of bias. And you can see that there's an awful lot of red and yellow on this graph. And so they detected a significant risk of bias across all of the included studies. And this was largely due to a lack of blinding and unclear methodological reporting. So as far as the reporting, we can fix that with complete transparency. But what about the blinding? In our conversation with the organizers, we heard that blinding is a common frustration of investigators in this field. If you think about it, trying to blind someone to a dietary intervention that they have to maintain is a little bit difficult. So what do you do in situations like that? Well, you can think about people at other parts of the study life cycle. So of course it's best to blind as many individuals as possible. But in addition to the participants and the physicians involved, you can also blind the data collectors, the outcome adjudicators, and the data analysts. But of course, sometimes blinding just isn't possible. What do you do then? Well then, of course, you want to try to standardize the treatment of groups as much as possible. Use objective, reliable outcome measures. Consider duplicate assessments, so have more than one assessor measure the outcome. But ultimately, acknowledge the limitations of the study so that other people can interpret the study correctly. But up until now, I've been discussing mostly human studies, but what about preclinical studies, like preclinical in vivo animal studies or in vitro studies? These in vivo and in vitro studies are pretty important for preclinical development, so you would think that their rigor and transparency is also important. So the question is, do animal studies transparently report these risk of bias items? And if not, is the lack of reporting associated with inflated effects, just like it is with human studies? Well, just like there are CONSORT guidelines for the human clinical trials, there's no shortage of various reporting guidelines to help with preclinical animal studies. These are some of the more general guidelines that aren't specific to a particular topic or technique, but they all aim to improve reporting of preclinical studies. However, despite this, there still seems to be an inadequate level of reporting. This is a review of over 3,000 preclinical cardiovascular studies, and the authors found that despite a small increase over time, only about a third of the papers reported whether or not they blinded, a quarter whether or not they randomized, and less than 10% included sample size estimation. And in another review, they looked at before and after implementation of a checklist at Nature Publishing Group that explicitly asks authors to report these items, and then compared them to papers in control journals that were outside of Nature Publishing Group. You can see in black is when they reported the item in detail, green is more of a passing mention, and white is, of course, when they didn't mention it at all. And what they found in sample size calculation, randomization, blinding, and data exclusions is that in all cases, there was an increase in reporting after implementation of the checklist, especially compared to the control journals. But in the end, only about 16% of papers in the Nature Publishing Group reported all 4 of these items. So it appears that checklists do help with reporting, but they're not a panacea for transparency. And just like in human studies, lack of transparency in preclinical studies can also affect the results. So this is a report of 24 preclinical stroke trials, and the authors counted how many of these so-called quality elements were reported in each paper. And so these elements include things like randomization, blinding, sample size calculation, and other controls. What they found was a negative correlation between the effect size and the number of quality elements reported. And none of the papers came even close to reporting all 10 quality elements. And so this suggests that inflated effect sizes can also be a problem for preclinical studies that lack transparency. And in addition to inflated effect sizes, lack of transparency and experimental design elements can also affect the likelihood of finding statistical significance. So this is a study that used over 2,000 untreated ALS model mice and then ran simulations to place their survival data into 2 groups, and then asked how often they saw a statistically significant difference in survival between the 2 groups. Now, since they're untreated control mice, any differences they find are a false positive. And what they found with 4 mice per group is more than 50% false positive rate. So more than half the time, they were seeing a statistical difference between control animals. But they were able to reduce that percentage of false positives when they took some confounders into account. For example, they took out the mice with a low copy number of the transgene because these mice just have a different baseline level of health. They took out mice with who died of unrelated causes to the study, for example, an infection. And they also implemented sex and litter matching. And they were able to reduce the false positive rate to about 30%. But they were also able to reduce the false positive rate by increasing the sample size. You can see here that when they had about 10 mice per group, which is fairly typical for preclinical studies, they still didn't reach the 5% false positive rate that people often think they have just because they're using a p-value of 0.05 as their statistical cutoff. No, instead they had to both increase the sample size substantially and consider these confounding factors to reach an acceptable false positive rate. Which brings me to the general point that null hypothesis significance testing and the p-value has its flaws. P-values were never meant to prove or disprove hypotheses or to tell us the magnitude or importance of our results. Instead, they simply show us the compatibility of our data with a particular model. And Ronald Fisher himself, one of the statisticians responsible for the concept of statistical significance, said in 1935 that if we accept this convenient convention, that p less than 0.05 is significant, we thereby admit that no isolated experiment, however significant in itself, can suffice for the experimental demonstration of any natural phenomenon, meaning a p-value in isolation doesn't tell us very much. And he also said that if the design of an experiment is faulty, then any method of interpretation which makes it out to be decisive must be faulty too. So we really need to pay attention to the design of our experiment and not just depend on the statistics. Now, in response to some recent abuses of the p-value, the American Statistical Association recently released a series of editorials in the American Statistician where overall they argue that scientists should move to a world beyond p less than 0.05. Instead of using a bright line of significance, they suggest instead that scientists are very clear about levels of uncertainty in their data as well as limitations in their study. I do, however, want to take a second to acknowledge the inherent difficulties in metabolism-based therapies research. It's heartening to know that there are researchers out there that are contending with these issues. For example, sometimes it's hard to get the sample size you need. It's hard to know what effect size to expect or to deal with unconscious biases, like how to figure out blinding in a study and when to include or exclude particular sets of data. But in addition, there's contending with baseline metabolic states and cycles like circadian rhythms, genetic and physiological differences across species, accurate measure of in vivo metabolites, and also figuring out what controls are most appropriate for a diet or metabolic intervention, since you're changing more than one thing at the same time. And it's not that researchers in other fields don't have to contend with these issues, but they are more acutely relevant in this field. And so these things just make it that much harder to do research in this space. And on top of everything, scientists are not performing their work in a vacuum. They're under various pressures. For example, there are professional standards that expect high quality research and a certain level of productivity. Journals often pit scientists against each other to tell the most exciting and novel story possible in order to even get published to begin with. Funding agencies and the peer reviewers there often take a scientist's journal publication record into account, as well as whether or not they've published in high impact journals to measure the scientist's quality. And on top of everything, institutional promotion and tenure committees heavily rely on publication record as well as funding history when deciding about career advancement. And so careful scientists can actually be punished in an environment like this. So all of these stakeholders need to take responsibility for building a culture of science that promotes rigor and transparency. And this is something we think a lot about in our office in particular. With that, I just want to leave you with some parting thoughts. First, involving research itself, just try to consider critical assessment of prior research, careful design and analysis of experiments, and transparency, transparency, transparency. So that way we know what was or was not done in any given study. And of course, help our colleagues prioritize these items. But also in terms of scientific culture, we need to increase awareness of the pressures out there that might be hindering our ability to do rigorous and transparent science. We need to build infrastructure that helps scientists do the best work that they possibly can, and we all need to work together. So all of the stakeholders in the scientific enterprise need to help foster a culture that promotes rigorous research. And to that end, our office has actually created a Slack workspace, you can see that here, where scientists of all levels, from trainees up to administrators, journal editors, as well as funders, come together and talk about best practices as well as new developments in this field. And if you'd like to learn more about that, just email us at rigorchampions@nih.gov. And with that, I just want to thank you for your time and attention. I have placed more information here of how to contact us or to learn more. But other than that, I look forward to our discussion in the live session.